Hello, everyone. I hope you're doing very well. I'm doing well on that end, and today we are going to describe muscle histology, the histology of the propulsion tissue, the propulsive tissue. We're going to describe, first of all, the various properties of the propulsive tissue. What are the characteristics of these muscles? Then we'll check on the various propulsive tissues. After that, we'll check on the functions of these propulsive tissues. Then muscle-associated terminologies. We also check the microscopic and functional anatomy of skeletal, cardiac, and smooth muscles. Then we'll also cover some of the revision questions. So kindly, let's do this. First of all, we define a muscle as any tissue that is responsible for the changes for the movement of the body or part of a body, and then that tissue will lead to change in size and shape of the internal organ. So, by the fact that that tissue is leading to one or two structures, one of the two structures will change in size and shape of the internal organ, that is a tissue, that is a muscle tissue. So, apart from talking about the various properties of muscle tissues, we need to understand that this muscle has to do a very special function, which is to alter the shape and alter the size of this organ of the body itself. So due to that now, you'll have the movement of the body or movement of part of the body. For the properties of the propulsive tissues, there are five. We have excitability, contractility, extensibility, elasticity, and conductivity. So for the excitability, this is the ability of the muscle to respond to chemicals released from the nerve cells. Those are the neurotransmitters now. So this muscle is able to respond to these chemicals. So it's able to respond to acetylcholine, glutamate, GABA, and other neurotransmitters. And the contractility property is the ability of the muscle or the tissue to shorten and generate force when it is under action, when it responds to these chemicals. Then it is its extensibility, its ability to be stretched without damaging the tissue. So it can extend or stretch without damage. And then elasticity is now after extending, it can back to return to its original shape after being stretched. And then conductivity is now its ability to propagate electrical signals over membrane. So now this one has to do with action potentials generation, hypopolarization, depolarization, and repolarization activities. So the first property can be able to be excited by responding to various neurotransmitters. And after responding to those neurotransmitters, neurotransmitters can cause this um, generation of action potentials. When it generates action potentials, enable the contractility ability of the propulsive tissues to generate force. Then, after developing extensibility, you'll have to contractility, you'll able to have the elasticity and the extensibility and the conductivity of the muscle properties. What are then these three types of propulsive tissues? So we have skeletal muscles, we have cardiac muscles, we have smooth muscles. Skeletal muscles are actually the most abundant muscles in the body. They're called epithelial muscles of the lower limb, muscles of the upper limb, muscles of the anterior abdominal wall, muscles of the neck. So talk about the sartorius, talk about the flexor carpi ulnaris, talk about the sternocleidomastoid, talk about the infrahyoid muscles, talk about the trapezius muscles. All those are just few examples of skeletal muscles. Then cardiac muscles, they are just muscles that are found on the heart. So cardiac muscles come from the word cardia, which is the heart now. So cardiac muscles, they are muscles that are found on the heart only. The smooth muscles, these are muscles that are found in three tracts. So they are found in the respiratory system, they are found in the gastrointestinal system, that is GIT, and then they are found in genitourinary system. So some of the muscles, trachealis muscle, we have trachealis muscles found in the trachea. Then you'll have the traxa muscle that is found in the bladder. You'll have various inner circular and outer longitudinal muscles that will be found in the gastrointestinal system, in the stomach. All those smooth muscles will be found in those tracts. Then what are the functions of these propulsive tissues or these muscles that you're talking about? So they carry out the movements, so they enable the body movements to happen. Then they stabilize body positions, they regulate organ volumes. For example, the stomach. So when the stomach is distended, food moves from the stomach to the duodenum. And also, when the food accumulates, when the undigested food materials accumulates in the rectum, the external and internal anal sphincter, they relax, they will relax and contract uh, at a certain circumstance to allow the removal of those materials via defecation. So regulating organ volumes is very important for the various sphincters, which are actually smooth muscles. So various sphincters, upper esophageal sphincter, lower esophageal sphincter, pyloric sphincter, cardiac sphincters. Talk about the ileal sphincter that will be found in the various locations, even including uh, the pancreatic sphincters that will be found on the second part of the duodenum, allowing the secretions of the pancreas into the gastrointestinal tract. And second that we have now, producing heat. So muscles undergo like continued contractions into these shivering movements to produce heat. In addition to that, we usually have some muscle cells which have contracted properties. One of them is the pericytes that are located in the blood vessels. Then you have the myofibroblasts, which are located in the connective tissue. To be specific, they are majorly found in the fibrocartilage. Myoid cells, which are found in the seminiferous tubules, they have that contractile property to allow uh, releasing of mature sperms. You have the myoepithelial cells, which are actually found on the exocrine glands, on the actually the beginning of the ducts to allow the release of those exocrine hormones or exocrine secretions into the target organs through the ducts themselves. So here's an example of a myoid cell that is found on the testes, the seminiferous tubules. So now this myoid cell contracts, it just enables the release of spermatozoa to move from, first of all, the periphery as they move towards the lumen of the seminiferous tubules, and then they are released to be stored or maybe to go to the vas deferens. So a myoid cell has contracted property. Then we check on the muscle associated terminologies. So a muscle cell or the muscle fiber is actually a myofibril. So a muscle cell at this point is a myofibril. Then sarcolemma is a specialized type of membrane that is found in muscles. So sarcolemma is just a cell membrane that is specialized and now called sarcolemma. It's called sarcolemma because it has infoldings. So these infoldings, they're the ones that make the transverse tubules. Then you have sarcoplasm, is actually specialized cytoplasm that is found in muscles. So this one is filled with the myofibrils and the myoglobuli. Then the sarcoplasmic reticulum at this point are specialized endoplasmic reticulum, which are enlarged periphery to form the terminal cisternae. And these terminal cisternae, together with these transverse tubules, they will make up the triad in skeletal muscles, and then we will make dyads in cardiac muscle. So the function of this sarcoplasmic reticulum is that it allows calcium control. So you remember calcium control is important in terms of generation of action potentials in cells. So three things can happen in terms of muscles. They can either undergo atrophy or hypertrophy. So two things, atrophy or hypertrophy. We can't talk about hyperplasia because hyperplasia is just division of the number of cells. But at this point, what happens to the muscles majorly is atrophy and hypertrophy. So in atrophy is the use or disuse. So if you use, you'll have hypertrophy. If you disuse, you'll have atrophy. So atrophy is a wasting away of muscles due to disuse or maybe absence of denervation. And this actually is even present in terms of paralysis. Then hypertrophy, you find that it is an increase in the diameter of the muscle fibers due to continued use. So for the people who usually go to gym, they increase the diameter of the muscle fibers, the diameter of the myofibrils. That is the reason why the muscles hypertrophy. They become bigger and bigger, the biceps enlarge. That is hypertrophy of the muscles. Then muscle as an organ, remember the criteria for any organ to be considered to be an organ, first of all, it needs to have a connective tissue covering it. It's also to have blood vessels in terms of arteries, veins, and it needs to have innervation. So a muscle as an organ, it contains connective tissue covering it. It also contains blood vessels in terms of capillaries, arteries, and veins. It also contains fatty vessels, and then it will finalize to have nerves for activation, or maybe the reason we'll talk about skeletal muscles are voluntary, cardiac muscles are involuntary. We'll see that in a few minutes. But remember, the connective tissue at this point is dense connective tissue. For the various muscle proteins, you'll have contracted proteins. These contracted proteins are the one that interact to enable now the shortening and the elongation of the muscles. So they are myosin and actin. And you'll see that myosin will form the thick filaments, and then the actin will form the thin filaments. So that is why so the contracted proteins. For the regulatory proteins, which now turn contraction on and off, so they regulate these contracted proteins. They are troponin and tropomyosin. So troponin and tropomyosin, they will regulate the active sites of actin towards ATP. So by displacing the presence of this tropomyosin, yeah. And then we'll also have now the structural proteins, which provide proper alignment, elasticity, and then extensibility. And they include titin, myosin, nebulin, and dystrophin, such as some of the structural proteins. And these muscle proteins now will bring, they are very important. That's the reason that some of them, for example, dystrophin, can bring a disease that is known as Duchenne dystrophic syndrome. So these are very important muscle proteins. I would like us to appreciate contractile proteins are myosin and actin, and regulatory proteins are troponin and tropomyosin. Structural proteins are chitin, myosin, nebulin, and dystrophin. For the connective tissue, at this point, we would like to talk about the connective tissue, which is, first of all, a dense connective tissue. So we'll start off with a single muscle fiber, a myofibril. So a single muscle cell, a myofibril, is covered by connective tissue known as endomysium. So endo means in. So covering the single muscle cell, the endomysium. Then you join various muscle fibers, will have a fascicle. So 10 to 100 muscle cells, 10 to 100 myofibrils will give you a fascicle. And then this fascicle will be covered by a perimysium. Then in this epimysium will be covering the whole muscle. Remember now that at this point, the muscle is being formed by many fascicles. So about 4 to various number of fascicles give you a muscle. which will be covered by epimysium. So this connective tissue at this point will be containing nerves, will be containing blood vessels, will also be containing some lymphatics. So that's the reason why the connective tissue is very important in terms of containing the structures seen. Then this connective tissue will not only be covering the muscle bellies, but also they will extend away from the muscle to form the muscle attachments. So in terms of the origin of the muscle and insertion of the muscle on bones will be due to presence of this connective tissue. So the tendons will be made by the presence of this connective tissue. So muscles attach to origins and insertions by connective tissue via fleshy attachments. A good example is the vastus medialis muscle, which attaches directly to the patella on the medial aspect. So connective tissue fibers are short. Now, for the vastus medialis, no, it does that not to even provide displacement of the patella. Then you have indirect attachments. So this one attach to form a tendon or aponeurosis. Some of the muscles that form tendons, we have the gastrocnemius, the soleus muscle, which form the tendon calcaneus, which attaches on the calcaneal tuberosity. Then we also have some other muscles. For example, you can appreciate the presence of the latissimus mastosi, which attaches via tendon on the floor of the bicipital groove. For the aponeurosis, the muscles of the anterior abdominal wall, external oblique, internal oblique, and the transversus abdominis, they attach the aponeurosis. And also talk about fascias, um, some of the muscles attached to fascias were friends of these indirect attachments. These are good examples. You can appreciate these are bone. So an extension of the connective tissue will give us a tendon. So this is indirect attachments. And then now the whole muscle will be covered by a connective tissue that is known as epimysium. And then this epimysium will be containing blood vessels, arteries, and veins. It will contain nerves, like it will contain some lymphatics. That's the reason you could appreciate this. The blood vessels, appreciate them there. Then inside of this epimysium, you'll find fascicles. So these fascicles will be covered by the endomysium. And then inside the fascicles, we have muscle fibers, the muscle fibrils, which will be covered by the endomysium, as seen there. So that is the connective tissues that are found in the muscles. I like us to appreciate that these skeletal muscles will be voluntary. So you've moved away now from the connective tissue muscles, then you're going to talk about the skeletal muscles. So these skeletal muscles are voluntary. So we have voluntary control. So they are not controlled by the autonomic nervous system, but you can be able to control them. So that's the reason why you can be able to move from one region to another by contracting uh, various muscles. For example, in your climbing stairs, you contract the gluteus maximus. Through that, now you can be able to move from one stair to another. Then these muscles are striated. These muscles are striated, and striations means we can be able to appreciate the dark and the lighter bands. And then these fibers are also long and cylindrical. The muscles or the cells themselves are multinucleate. And then in addition to that, the nuclei are peripherally located. And that can be well appreciated in this next image. As you can appreciate these are three layers of fibers, three fibrils, and appreciate presence of peripherally located nuclei. So peripherally located nuclei. In addition to this, we can appreciate regions of darker staining zones and regions of lighter staining zones. So this is lighter staining, then this is darker staining zones. Then you have multinucleation that is present in skeletal muscles. Then you can also be appreciate that these fibers are parallel to one another, they are long, and then they are cylindrical in nature. So we can't find branching. They're not branched. They're non-branching. And then another point to appreciate is that they have multiple nuclei because each cell was formed. Each cell was formed by fusion of about 100 embryonic cells. So 100, more than 100 myoblasts fused to give us one long and multinucleated myofibril, one multinucleated cell. And we can actually appreciate that in this, you can see these are the myoblasts. Then these myoblasts, they became the satellite cells. Then the satellite cells, they fused to give us a myofibril. That's the reason why the nuclei, first of all, they are multinucleation, one on the periphery, and then you can have regions of dark and light staining zones. So these are an electron microscopy of the same. Remember, for this, these are just like microscopic features that you can be able to appreciate. But now we would like to appreciate what makes these dark zones and eye zones. Those are the situation that we're talking about. These dark zones and light zones, what usually form them. And we can actually appreciate that from a muscle, you have a fascicle. And from a fascicle, you have a myofibril. And for this myofibril, we like to appreciate what are these regions which are darker staining and what are these regions that are lesser staining. We say that the functional unit, of a muscle is a sarcomere. And that functional unit of muscle is a sarcomere. It's actually the regions between two Z lines. So this is Z line, this is another Z line. So the distance between two Z lines make up sarcomere. And we have a zone that is known as A band. So the A band is a region that is the dark zones that you see here. So the dark zones here, these dark zones at this point are the A band. And then the lighter zones are the I band. So you should have two bands, dark bands, and lighter bands. So the lighter bands are the I bands, the darker zones are the A bands. So the A bands are actually made up by the myosin filaments. The I bands are made up majorly by actin filaments. So in between now the A band, we have a H zone. And this H zone at this point will be containing a region of lesser, but like it's not darker staining. So this is I. This is a high H zone. And then the H zone at this point, it will be containing an M line in it. It will be containing an M line in it. But a point to appreciate is that in between Z line and the A band, we have a region of I band. Have a region of I band. So this M line is an I band, this is the I band, and then this is Z line and this is I band. So the sarcomere is a distance between two Z lines. So this is a Z line, it's another Z line, and another is the sarcomere. So the sarcomere will be containing the A band, two I bands, a H zone, an M line inside there, and then you also have now the biopsy, the actin, and the various fibers containing there. So this one is an electron microscopy features of skeletal muscle. So if a question can come at this point, you can be asked, describe the electron microscopic features of the skeletal muscle. We're supposed to cover the sarcomere, the I bands, the A bands, the H zone, the N lines, the Z lines, the myosin, the actin, the troponin, those structures there. And then at the end of this class, I'd like us to go and draw this sarcomere and be able to label all those zones that I talked about. Before we move on, I'd like us to appreciate the presence of this sarcoplasmic reticulum. So this sarcoplasmic reticulum, together in the terminal system line, they form the triad of the skeletal muscles. So that's basically about it. Can appreciate the thick bands are actually the mousing, the thin bands are the actin. What about now cardiac muscles? So cardiac muscles are they have involuntary control. Remember how skeletal muscles say that they have voluntary control. So this one have involuntary control, and they can be able to initiate their contractions by themselves. So they have autorhythmic cells. They are autorhythmic cells. They can be able to contract and relax without nervous stimulation. And they are striated, same case as we saw in the skeletal muscles. Striated means they have regions of fiber, region of dark zones and light zones. Then they are short, they are quadrangular in shape, and then they have branching fibers. So if you can be able to contrast now between skeletal muscles and cardiac muscles, we find that skeletal muscles are long, cardiac muscles are short, cardiac muscles are quadrangular in shape, skeletals are symmetrical in nature, then you have branching pattern that is present in cardiac, of which it's absent in skeletal muscles since they are parallel to one another. So here you can appreciate cardiac muscles, you can appreciate that they are branched. This is a branching that is present there. You can also appreciate the branching that is on there. Then you can appreciate that these muscles are short. They're short and somewhat quadrangular in shape. Then they have single centrally located nuclei. So these are nuclear. So they have mononucleation or binucleation. Mononucleation or binucleation, those is mononucleation. That it's and then the nucleus is actually located at the center of the muscle cell. So these are muscle cell, the myocyte, and you have the nucleus before at the center. So you'll find that this cell will be somewhat circular in nature. Then cells connected by pairs of intercalated discs. So these are the various intercalated discs. They are darker staining. It's a darker staining intercalated discs. So these intercalated discs, they just enable communication of one muscle cell to another via gap junctions, focal adherence, and desmosome. So they have three junctions: gap junctions, focal adherence, and desmosomes. But the most important junction is gap junctions. And then they have same arrangement of thick and thin filaments and skeletal, giving us presence of the striations. But now at this point, we'll be able to appreciate the branching pattern that is present. So this image shows us the centrally placing of the nuclei. So we can have mononucleation or the binucleation of cardiomyocytes. Then these muscles are quadrangular in shape. You can appreciate them somewhat quadrangular in shape. They are short. They are joined by an intercalated disc. So some is an intercalated disc, another one is here, intercalated disc. And then we have the branching pattern. So you can see desmosomes, you can see gap junctions, and you can see focal adherence. And of course, you can appreciate presence of capillary because the heart has blood supply. Then you can appreciate the sarcolemma that is present. Another feature is that they have increased number of mitochondria. Because of that contractile, they require energy. For smooth muscles, as well, you find that they are spindle-shaped, they are non-striated, so they would have the regions of dark zones and light zones. Then we'll have the central nuclei, which actually be found at the center. And then these muscles will be arranged in sheets, in layers around the tube, for example, around the esophagus to allow some movements like peristalsis to happen. So through that now, they can be able to keep contractions going for a longer time. And then in addition to that is that we find muscles can be able to contract and relax at various points of the tube. So that due to that now, you can be able to resist fatigue to prevent accumulation of lactic acid. And then also you can be able to control the muscles. So remember, these smooth muscles, they are not under voluntary control. They are actually under involuntary control. So opening the sphincters, relaxing the sphincters, contracting the sphincters, allowing the various peristaltic movements to happen, major migratory complex movements of the large intestine to happen, can be due to unconscious control. That is autonomic nervous system, stretch, and even hormones is happening. So pressure of food in any tube, in the stomach, in the duodenum, we like to stimulation of the nervous system, and through that now you'll be able to move and have various movements happening. Not only in the GIT, you can also have movements that are happening in the genitourinary system, movements that are happening in the respiratory system under the influence of autonomic nervous system, stretch, hormones, and even some by drugs. You can appreciate these as spindle-shaped nuclei, the spindle-shaped cells. These are nuclei that is centrally placed. So this muscle, you can find them in the bladder, you can find them in the uterus, you can find them in the esophagus, you can find them in the trachea, you can find them even in the urethra because they have that function. Then a good example is this. You can be able to appreciate presence of now the spindle-shaped nuclei that is present there. So that is it. You can appreciate the circular muscles, the outer longitudinal. So inner circular, smooth muscles, inner longitudinal, smooth muscles. So the cross section actually is well found is that for the inner circular, you can appreciate how the muscles are transverse. And then for the outer longitudinal, you can appreciate the centrally located nuclei in these smooth muscles. Then you can now narrow down to various. So a brief recap is that skeletal muscles are found attaching to various bones. Cardiac muscles are only found in the heart. Smooth muscles are found in the very tracts, the respiratory system, respiratory tract, the genitourinary system, and then also in the gastrointestinal system. So some few questions, identify the tissue shown and give 3 reasons. We can appreciate that this is skeletal muscles. Why? Well, these skeletal muscles can be able to appreciate that the fibers are parallel to one another, can appreciate the nuclei are found on the periphery, can appreciate presence of striations, can be able to appreciate the fibers are parallel to one another, they are somewhat cylindrical in nature, and you can also appreciate presence of multinucleation. So list 3 ultrastructural features of this tissue. So these are the features that you described, features that are found in the sarcomere. So all the structures that we talked about will be listed there. Then what is the functional unit of this tissue? We've talked about it. This tissue is skeletal muscle. It will contain a sarcomere as a functional unit of this tissue. Then you have this other structure. So I can give you 2 minutes to answer the question. I believe you've been able to answer the question. So this one at this point is not a muscle. This one is dense, regular connective tissue that is present in tendons, ligaments, and aponeurosis. You can appreciate that. So ideally, you can appreciate that this fiber first of all, yeah, it's more fibrous than you can appreciate cells in lacuna and examo. Sorry, you can appreciate that it's more fibrous as connective tissue, but ideally you can't appreciate the peripheral nuclei. You can't be able to appreciate the striations in this. So absence of striations is dense, regular connective tissue. Then you have this other one. I can give us 1 minute to answer it. I believe you've been able to identify this as smooth muscle. Can appreciate the presence of spindle-shaped cells with nuclei located at the center, and then ideally they're found in layers. They look like sheets of cells laying on one another. And actually that is well depicted in the image that is just next there, can be able to appreciate uh, these are the inner circular muscles, the inner circular muscles, and then outer multiple muscles. So presence of small muscles, ideally, can appreciate the sphenoid cells. Then for this other one, you can give us 1 minute also to discuss about it. So I presume you're able to talk about that these skeletal muscles can appreciate the striations, very prominent striations. Then you can even appreciate the presence of connective tissue, the endomysium at this point. Then you can also be able to appreciate the nuclear fauna on the periphery. Then you can also be able to appreciate the fibers are parallel to one another. But then there is no branching, cylindrical in nature, and then fibers are long. For the next one, you can be able to appreciate that this is actually cardiac muscle. So you can be able to appreciate, first of all, the nucleus is actually found at the center. You can be able to appreciate the branching pattern. You can be able to appreciate the presence of intercalated discs. There is an intercalated disc there. Another intercalated disc is found here. Another intercalated disc is found here. Regions which are darker staining, another intercalated disc. Then you can be able to appreciate binucleation. Multinucleation is present in, so you can appreciate centrally located nuclei, centrally located nuclei at the center. Then you can also be able to appreciate that these fibers, these muscles, the cardiovascularies themselves will be highly branched. They have presence of striations, can appreciate striations being present. And then you can be able to appreciate presence of intercalated discs. So remember, these intercalated discs will be containing the gap junctions majorly, desmosomes, and focal adherents, enabling now flow of calcium from one muscle cell to the other, now enabling the muscles to have flow of action potentials from one muscle to the other via the sarcolemma. And through that now you'll be able to make the muscles to contract as a functional unit, as a functional syncytium. So that is a cardiac muscle. We can be able to appreciate now the other one at this point. This one should be simple at this point. This is skeletal muscles with the various characteristics that we talked about. So it's skeletal muscles. Be able to appreciate the periphery located nuclei. At this other point, I would like us to appreciate this is, maybe I can give you 1 minute to discuss about it. So this is cardiac muscles. Cardiac muscle can be able to appreciate presence of striations. You can appreciate striations. So when you see striations, you eliminate smooth muscles. You remain in skeletal and cardiac muscles. If you appreciate the presence of cardiac intercalated discs, you also be able to appreciate the branching pattern that is present. That will give you now a characteristic of cardiac muscle. So remember the other characteristics that I talked about, peripheral, centrally located nuclei, being able to appreciate that the nuclei, they are somewhat rounded, they are not flattened, and then be able to appreciate even that the cells, they are short and then quadrangular in shape, and then they are highly branched. For the other one, I can give you 1 minute to discuss about this, but just figure out this. Ideally, is it a muscle? Is it a cartilage? Is it a connective tissue? So is it a muscle? I presume you've said no. Is it a cartilage? I presume you've said yes. Which type of cartilage? Elastic, hyaline, or fibrocartilage? I presume you've said. fibrocartilage. So you can appreciate it's highly fibrous. Then you can also appreciate the presence of chondrocyte cells in lacuna. So at this point, I'd like us to go and revisit the video that we did on cartilages. This image has been well described in that video. For the other presentation, we can be able to appreciate the presence of centrally located nuclei, centrally located nuclei, centrally located nuclei, and the centrally located nuclei. And you can be able to appreciate the striations, the branching pattern that is present, appreciate the branching pattern that is present. Then you can also appreciate that these nuclei, they're not found on the periphery, and also they're not flattened. They're not flattened. These are just muscle fibers that have been destroyed during sectioning of the slide, but these are not nuclei that are found on the periphery. So you can appreciate nuclei that are actually found on the centrally located nuclei, centrally located nuclei, centrally located nuclei, being able to appreciate the presence of intercalated discs here. Another intercalated disc is present here, another one is here, another one is here. Those regions of darker staining zones, which are the intercalated discs. So this one is a cardiac muscle. For the next slide, I'd like us to describe what you can see in our screens. At this point, I presume you will say that this is a skeletal muscle, first of all, a transverse cross-section of skeletal muscles. The other ones that we've covered, they're just longitudinal cross-sections. That's the reason we are able to appreciate the striations. The most important thing for us to appreciate is the presence of, first of all, the endomysium covering a muscle fiber, and then the nuclei found on the periphery, and then the multinucleation is also present at this level. Then you can also appreciate the presence of blood vessels in between the various muscle fibrils. I presume the last slide, I'd like us to appreciate that this, is it a muscle? Is it a cartilage? Is it a connective tissue? So first of all, this is not a muscle. It's a cartilage. Which type of cartilage? It's fibrocartilage. To identify the tissue and give two reasons, you can be able to talk about that this is fibrocartilage. You can appreciate cells in lacuna, the chondrocytes in lacuna. Then you can also be able to appreciate it's highly fibrous in nature. Then the reason why it's not a muscle is it doesn't have striations. Then the collagen fiber that is abundant throughout life is collagen type 1. And for the distribution and function of it is that you find it in places where you require support. So in the intervertebral disc, the annulus fibrosus, the pubic symphysis, the symphysis menti, the temporomandibular joint, the sternoclavicular joint, and then you also find it in other specialized types of joints. So those are the differences between skeletal and cardiac muscle fibers. We can take our time and go through them. Maybe you can take a screenshot of the slide, then you go through it at your own time. Be able to differentiate between size, length, shape, and position of the nucleus, striations, cell junctions, and then you talk about the positions of T-tubules, tired, stroke diad, mitochondria, innervation, contraction, and even regeneration. So those are some of the questions that you might be asked. And then I hope you really enjoy the session. May the Almighty bless you. And then this just a reminder that you shouldn't watch the clock, but you should do what clock does. So you keep going, you keep moving, you keep working, you keep reading, and God will always bless the work of your hand. So with that, have a good time, all the best in your studies. I wish you all the best in your endeavors. Thank you.